Birgitta Duim. I'm an assistant professor at the Faculty of Veterinary Medicine of Utrecht University in the Netherlands. And it's my pleasure to talk about the work we do on plasmids in Campylobacter. And this is mainly work from my colleague, Linda van der Graaf. She's a postdoc in my group and she did all the nice work on developing a typing tool for Campy also. So Campylobacter is associated worldwide with gastrointestinal diseases, and genome sequencing has become very popular for studying the epidemiology of Campylobacter, especially the Duim and coli. But plasmids have not well been characterized yet. So that was our aim, to look at the diversity and the content of plasmids in the whole phylum of Campylobacterota and develop a typing tool. And for this we had two data sets with genome sequences, plasmid sequences from public databases. And for the taxonomic classification, we had a data set with plasmid sequences from different species within the Campylobacterota family. And for the typing tool, we specifically had genome sequences from Campylobacter jejuni and coli. And we also sequenced 13 strains with long-read sequencing for Campylobacter jejuni and coli, and both were around 1,100 sequences. And when we then cluster the plasmids using the so-called an ACC network partite network, you can see here that's based on the homology between the plasmid-encoded proteins. You can clearly see that the plasmids of Campylobacteriaceae cluster separately within the whole phylum of other species within the Campylobacterota. Also, when we then cluster the plasmids based on the average nucleotide identity scores, which groups them into so-called plasmid taxonomic unit clusters, you can also see here that there is a clear differentiation of the Campylobacter plasmids. There are 13 different clusters with Campylobacter plasmids and 5 with Helicobacter plasmids. But there are also gray clusters which couldn't be classified at all with PTU clustering. And this really improved when we used the characterization of 4 marker genes, and they are part of our typing scheme. They consist of genes involved in replication, immobilization, and the genes for single binding proteins. And also from the types 4 secretion system, the 4D gene is included. Using these 4 marker genes, we clearly differentiated more Campylobacter jejuni and coli in separate clusters. And that's shown here in the bottom in the figure, where we have the phylogeny based on the gene presence absence of these genes. And you can see here in the inner circle, where in yellow there are the Campylobacter jejuni and coli, and in the other colors there are the other Campylobacter species. And what I want to really show you is that there is really here a large cluster with a similar cluster with the similar plasmid. And this plasmid is carrying the resistance genes, the TET-O for tetracycline resistance and the HPA3 encoding aminoglycoside resistance. So this is clearly an indication of the similar plasmid that is present in jejuni and coli, but you can also see that it's present in other Campylobacter species, in Nova Zealandia, in Vulpris and in Uptaliensis. So a clear indication for horizontal gene transfer of AMR genes between Campylobacter species. So in overall, we see that there is a good separation of Campylobacter plasmids in the whole phylum of Campylobacterota, although with different resolution, whether you use ANI or protein homologies. But the typing tool really improved the differentiation of Campylobacter jejuni and coli. And that typing tool will become available in an Apricot tool, so that everybody can use it for their own sequences to do the analysis. So when there are further questions of this research, you can contact my colleague, and thank you for your attention.